CRISPR-Cas gene editing technology is a groundbreaking tool in genetic engineering and molecular biology, known for its precision, efficiency, and versatility. Here's an overview of its components, mechanisms, applications, and ethical considerations. Let's talk about components of CRISPR-Cas genome editing technology. 1. CRISPR, abbreviated from clustered regularly interspaced short palindromic repeats, CRISPR, are segments of prokaryotic DNA containing short, repetitive base sequences. These sequences are derived from DNA fragments of bacteriophages that had previously infected the prokaryote, used as a defense mechanism. 2. Cas proteins. CRISPR-associated proteins, Cas, such as Cas9, are enzymes that use the CRISPR sequences as a guide to recognize and cut specific strands of DNA complementary to the CRISPR sequence. And 3. Is guide RNA, gRNA. The gRNA is a synthetic RNA sequence that matches the target DNA sequence. It guides the Cas9 protein to the precise location in the genome where the cut is to be made. Now the mechanism of action is as initially. Initially binding happens where the GRN-Cas9 complex binds to the target DNA sequence via complementary base pairing between the GRN and the target DNA. Then the cleavage stage, where Cas9 introduces a double-strand break, DSB, in the DNA at the target site. And lastly DNA repair stage where the cell's natural repair mechanisms, such as non-homologous end joining, NHEJ, or homology-directed repair, HDR, then repair the DSB. It often results in insertions or deletions, indels, which can disrupt gene function. HDR can be used to introduce specific genetic changes if a repair template is provided. Now let's talk about the applications of CRISPR-Cas technology. They have several applications in different fields, for instance. In biomedical research, CRISPR is used to create model organisms for studying diseases and genetic functions. It also enables high-throughput screening for drug discovery and functional genomics. In medicine it is used in gene therapy, correcting genetic mutations in diseases like cystic fibrosis, muscular dystrophy, and sickle cell anemia. Cancer treatment, engineering immune cells to better recognize and attack cancer cells. In agriculture, CRISPR-Cas technology is used for developing crops with desirable traits such as drought resistance, pest resistance, and improved nutritional value. And in synthetic biology, this technology is used for creating organisms with novel capabilities for biotechnology applications, such as biofuel production, and environmental remediation. There are several ethical considerations. 1. Safety. Off-target effects, unintended edits to the genome that could have harmful consequences. Long-term effects, potential unforeseen consequences of gene editing. 2. Accessibility and equity. Ensuring equitable access to CRISPR technology and its benefits, preventing a divide between those who can and cannot afford it. 3. Germline editing. Editing germline cells, sperm, eggs, or embryos, can result in heritable changes, raising ethical concerns about consent, the nature of human genetic modification, and potential for eugenics. Now let's have a look at future directions. 1. Improving precision. Developing more accurate and efficient Cas proteins and delivery methods to minimize off-target effects. 2. Expanding applications. Exploring new applications in diverse fields, such as industrial biotechnology, environmental science, and personalized medicine. 3. Public engagement and policy. engaging with the public to discuss the implications of CRISPR technology and shaping policies that reflect societal values and ethical considerations. So summarizing, CRISPR-Cas gene editing technology holds immense potential for scientific and medical advancements, but it must be pursued with careful consideration of ethical implications and societal impacts.